my pleasure to introduce Dr. Amani as an invited speaker to the session. Dr. Amani is currently an associate professor in the Department of Biotechnology and Engineering and the Deputy Director of Innovation and Commercialization at Research Management Center, International Islamic University, Malaysia, Kuala Lumpur, Malaysia. Dr. Amani received her BSc and MSc in Electrical Engineering from University of Minnesota, Twin Cities in 2001 and 2003, respectively, and her PhD in Biomedical Engineering from Purdue University in 2009. Prior to her position at IIUM, where she is presently teaching and mentoring research students, Amani was a researcher at Purdue University, USA, and was appointed as a principal investigator to a NASA small satellite project in 2012 to 2014. I welcome Dr. Amani to the international conference IconMesh 2020.
Dr. Amani is not going to present online. Her video will be played.
Okay.
Good evening, everyone. I'm Amani Salim. I'm from the Amani Research Group. I'm the principal investigator of Amani Research Group. Amani Research Group is part of the Nanoscience and Nanotechnology Research Group at International Islamic University, Malaysia. First of all, I would like to say thank you to organizers for inviting me to give a talk to this conference, very important conference, because of the interdisciplinary nature of the conference itself, where it's nice as an engineer scientist to share my work with other communities, such as social science, management, and humanities. So I'm grateful for that, and thank you. Maybe there could be cross-pollination or collaboration between these fields if we find ways to collaborate in such endeavors. So anyway, my title of my talk today is about nanomaterial-modified electrodes for biosensing. Before I go further, I'd like to give an introduction to what is a biosensor. So the concept of biosensor can be described in this figure. So it consists of three distinct components, which is a receptor, the interface layer, and also the transducer. So if you take an example of a glucose sensor, you have this something that you targeted to detect, which is the biomarker, which is identified by these antibodies on these electrode surfaces. And of course, it's amplified by this transducer to basically give you a signal from the interaction between the target and the receptor, which we call recognition layer. So for the past 10 years, my group has been looking into how do you improve the performance of such sensor. In this case, we took a case of an electrochemical sensor because it is scalable, it's readily available, really suitable for a lot of developing countries' labs. You don't need fancy equipment and so forth to develop these sensors. However, they have limitations. One of the things is that if you look into the typical glucose sensor, okay, you look into typical glucose sensor, they have all these layers that they put on the electrode surfaces, starting of course with the membrane itself, so that you can select the targeted analyte in the recognition element. You have all these layers of electrolyte, and then of course all these signals that happens in the biochemical domain is transduced to the electrical domain, number 5. However, from these, of course, all of these layers require processes, and this is a glucose sensor. So we're able to only detect glucose sensor from samples such as blood. And the thing is, in this domain of sensors, is that instead of blood, can you go to much diluted samples, such as is there glucose can be detected besides blood, but also in other bodily fluids such as urine, saliva, sweat, or tears. And besides that, we use a glucose sensor, as you can see here. It's a typical glucose sensor strips that the signs have not been, there's no innovative science has been done on the glucose sensor strip space. There's a lot done in the monitoring space, but in terms of the strip science, it remains as is. There's not much innovation if you look going on in the space. So what my group is doing is that some of the projects I work on are very fundamental, but then some of the projects I work are also to commercialize a lot of my work by taking the low-hanging fruit of things such as how can you make a glucose sensor to be able to detect glucose besides in blood in modality samples such as saliva. So because of that, for the past 6 years or even longer than that, even when I was in the United States before I come to Malaysia at International Science in Malaysia, we work into looking can we improve this transduction by using nanomaterials. We try to understand transduction of the electrons through modification of the nanomaterials transducer, with of course the presence of recognition elements such as for the glucose sensor is glucose oxidase. For immunosensor, of course, we use antibodies and so forth. And we try to understand the complex relationship between all the experimental parameters involved in nanomaterials by using machine learning techniques and machine learning to model data and to understand the interaction between input and output for the sensor. So if we change in nanomaterials, how does the change in nanomaterials affect the sensor performance? So something that we work on to look into the several parameters involved in the glucose sensor space, which is whether the system is reversible or not, because reversibility of system affect whether the transducer of ECAM sensors can basically transduce electrons at the electrode and solution interface. We look at the electrons transfer, mass transport, effective surface area. High surface area, of course, allows for more anchoring of biomolecules on the biosensor surface, therefore enhancing its performance. So something that we look at for the past several years. Besides that, we also look at, of course, a lot of this ECAM sensor deals with redox reaction, transport of ions to nanomaterials. So we look at several nanometers electrode surface. We look at materials such as conductive polymers, interaction with nanomaterials and so forth, and how it affects, of course, the selection of the analyte, which is in this case ions, and how does it enhance the electrons transfer by the surface. So we do a lot of modeling and so forth, based on fundamental work of known scientists who look into these fields, such as Joseph Wan or Mobaka, in this case, who study interface between electrodes and also, of course, the solution. And because of that, we managed to develop, we also look into the high priority areas, and I said earlier, one of the non-invasive diabetes monitoring by measuring glucose in saliva, and because this is a multi-billion-dollar market, so we focus on that, especially with developing countries. Our strategy of doing things has to be very lean. So we look, this is a low-hanging fruit that we can use some technology to create a multi-billion-dollar business. At the same time, besides glucose sensor, we also can apply the same technology that was used in the glucose sensor space into the environmental monitoring, remote security and defense. So throughout the years, this is what my group, which I call the Amani Research Group, our group has been working on sensors technology. And if you can see here that our group has worked on a lot of improving detections of sensors technology, not just calcium in this case, carried with that, but also uh, things that evolve from standard lab testing into something that which is improved through methods of biochemical methods, such as ELISA and so forth, into more of using and manipulating nanomaterials, such as graphene and kinetic polymers, to create sensors technology. Some things that I was inspired as my work at NASA, images calcium in space. We use the same sensor technology to develop all solid state electrodes for water monitoring and also for calcium sensing for detecting early pregnancy complication by looking into the calcium intake in embryos. So we also look into that. Just to show you the work that I did at NASA. So this work that I did at NASA. So some of the work I did at NASA was to use these ECAM sensors in this compact case to measure extracellular calcium ions from the single cells. This is a furnace bore cell that we put in between the electrodes. So we modify these electrodes with a simple silver chloride layer. Positioning of the cells, close proximity to the electrodes actually affect the sensor performance. So we did this in a spaceflight experiment and learning from my work at NASA, we developed new materials, of course, to develop sensors for the ground application. In this case, as I told earlier, it's not sensors. We also work on, as I said earlier, application in determining the concentration of calcium ions in biofluids, and it has to do with also to differentiate between early complications in pregnancy phenomenon and pregnancy with complications and so forth. So we're looking into more on the women's health space, whether how the sensors can be applied for women's health. We also use this for a lot of projects. It has the attention of a lot of people in the water space. So especially when there's issues of water pollution that arise due to chemical dumping and so forth, also occurs in Malaysia, just increased demand for the freshwater supply and so forth. Therefore, monitoring technologies of our rivers, not just rivers, but also of the water source throughout Malaysia, even the marine life and so forth, requires use of sensors technology. Therefore, we also focus on developing a mobile sensor for such technology, basically to study nanomaterials and see how can enhance performance of these sensors. So we developed this mobile sensors technology where we studied the sensor performance and then have it deployed in rivers so that it can detect the water quality in the rivers using the nanomaterials or position in a certain structure to enhance the electrical consumption on the electrode surfaces. Besides that, we also use for these biosensors to handle emergency situations. One of them is the first time that we developed these sensors was used for the detection of E. coli in water, especially in areas in the east coast of Malaysia where there's occasional floods and so forth. So we use it in disaster areas. We work with NGOs and so forth to develop technologies that can go in the field and can be used by the communities. We of course quantify how the sensors behave with the concentration of bacteria and so forth. And these biosensors can give a turnaround time of 120 seconds. You can tell basically quantity of the electrode of the bacteria on the sensor surface and quantify it faster than if you send it to a biochemistry lab. So the value proposition for all of these sensors is that they low cost, you need a microfabrication cleaning facility, easily deployed for emergency situations. And most of our are already at the TRL 4 to 6 because this prototype has been verified in the field, especially in the east coast of Malaysia. Finally, last but not least, we also apply a lot of this technology to develop new things. One of them is a smart bandage, just interfacing these sensors with the nanofibers. So we develop also these nanofibers in the lab to create a smart bandage, basically to monitor wound healing. So these sensors are also adapted because of the nanomaterials can be adapted in smart bandages technology, which is really nice. So anyway, giving you the overview of my work, I hope that I have shared a general overview of how electrodes that are modified nanomaterials can be applied in applications in glucosensing, space biology, can be used in water quality monitoring, also in smart bandages for wound healing. To give a special thanks to especially our group, of course, is small and mighty. The nano research group, of course, is larger. Group consists of many more lecturers, but in my small group, I have my research officer here on the left here, Pirabin, and then Hussein from Algeria is my first PhD student in International Science University in Asia. And this is me, and here's Uswa. So Pirabin is working on the smart bandage, Mohsin is working on the quality, Uswa is working on the biomedical applications. I have to say thank you to our industry partner,